All right. So sorry, I'm sure. I'm not sure I'm hungry.
Hello, hello. Okay.
Oh, this works very well. Sound was very clear.
Yes. So I just turn it on and you can press this.
Okay, good. The laser pointer. Okay, all right. I can use the laser pointer. It's moving too fast. Okay.
Sure, this is John Potempa. Yeah, I'm not just Kim. Kim is the director of CBR. He'll be meeting you at about 4 o'clock this afternoon around that. So after this talk, I think come and talk and take him down, to which will be detailed, and then you should.
I look forward to seeing your profile. Thank you for coming.
He's a very honest, so you'll get something out of it. You actually practice 1 day a week sometimes.
Yeah, that's where it was yesterday. Unfortunately, I could not attend the trial because that was I was seeing patients. Dealing with patients.
In a very direct manner. Through the translation.
It's a different angle.
Thank you very much.
Thank you. Writing was great. Yeah, very well. I will take him down. I will take him down and bring him back. Oh, yeah. Yes. Okay, looking at you get come to dinner. I don't know. Okay. Should we bring after the yes. Most of my friends. Okay, so I will be again. That's all. And I'll tell you the text.
Yes, I know. I'm stealing the water from the hotel, so probably I will have it too.
Okay. All right. So,
Mhm.
Can you pronounce your university?
Università Jagiellonia, no, Jagiellonia.
Okay, I'll get you to say it.
Jagiellonia.
And,
It's here, it's written here, so
I
you
still
can,
can't pronounce it.
Jagiellonian University.
And that's so you went to Jim Travis in Kentucky,
In Athens, Georgia.
in
Athens, Georgia, University of
Athens, that's
Georgia.
right,
Yeah, in Israel.
as a postdoc.
As a boy.
Right, and then you got your position.
In 85, I went there and then I became until moving to
Right, and then at the same time, you're able to get a position
in Poland.
at the same time.
You know, saying all the time. I was a
Even
position.
whilst a postdoc.
Yes, even as a postdoc. As a postdoc, I was delegated to do the postdoc at the time.
Okay.
So I was on the payroll.
Oh, I see.
Then they changed the rules in Poland, and so now I can do
Well,
the
that was before the war came out.
That did, yes, yes. Exactly.
Yeah, so that's
I
how
remember
bad
being
it was.
when the war went down.
So you actually were in did the war come down when you were in Georgia?
In Georgia. So I was in Poland at that time, so I was going back and forth in Poland.
Yeah.
First time I was 85 to 87, then 89 to 19. Then, every I was playing back and forth at that time.
So what is the super elite level 100,000 member in United Kingdom?
1K, okay.
1,000K.
I
Which is
don't know,
stupid.
1K.
1K. What does it mean? What is it?
I don't know. I think it's 1 million.
Yeah, exactly.
Like that.
Yeah. 1K doesn't make any sense.
Doesn't make any sense. But
Should be
it is the highest,
Yeah.
honor. So you have the special check-in actually for 1K in the airports. And so in business, they go to K1 to check in, and also you upgrade to business class. And then right now, you can always upgrade to business class, which is great.
Oh, that's nice. That's nice. And just lastly, when did you get those two awards, those two prizes?
It was in 2000, 2020, and 2019 and 2021.
Okay, great.
Hi, so it gives me great pleasure to introduce to you today Professor Jan Potempa, whom Dieter and I have known for about 26 or 7 or 8 years. As you can tell from the same color hair and some of the lack of it. Yeah. Dieter and I got to know Jan through the protease community, and Jan has got a large and long history of significant papers, over 500 papers, and giving him an H index of 90. And so for the summer students and whatever, an H index indicates 90 papers of over 90 citations in that case. So it indicates that his work is not just read, but it's cited, and it means he's also influential in the field. So this is great. And he's also recognized as being the world's leading expert in Porphyromonas gingivalis. So this is an oral bacterium that you're going to hear about, but also has a lot of systemic activities that Jan has been driving the field in as it leaves from this normal organism in the mouth and goes to heart or brain. So Jan has this interesting distinction of coming to the States, to Georgia in Athens, to do his postdoc whilst at the same time as being a professor at the University of, I can't pronounce it.
Jagiellonian University.
Yeah, way too many consonants, right? That university. And so he was there and they encouraged him to do a postdoc, which was great. So he then went to Georgia to Jim Travis, who's one of the great pioneer chemists in proteolysis. And from that experience, he then was able to complete that and then get a position also in the States at Louisville in Kentucky. So he's also unique in having 2 positions simultaneously, double teaching load and so on, but with some advantages. So he's got labs both in America, and so I can get NIH grants, and also in Poland, in No. The town is Krakow. Yeah. Sorry. And so it's also significantly that you're going to hear about today is work that you can see why he was given these two great awards in 2019 and 2020, and these are considered like the Nobel Prizes of Poland. So not international, but within the country. So the two top science awards. So he's highly decorated, highly recognized, highly published, and it's a really great honor to have him here today. And we look forward to your talk, Jan. Thank you very much.
Thank you very much for such a kind and flattering introduction. I am very happy to be here. And no, okay, it was not. Everything was true, actually, so I can justify that. All right, so but I'm very happy to come here to Vancouver, and thank you for inviting me. So I'll be talking today about applying Koch postulates to prove that specific bacteria is responsible for a disease. In this case, we'll be applying these Koch postulates to bacterial pathogenesis of Alzheimer's disease. As you see, this is my affiliation, Louisville, Kentucky. Then you see in Kentucky, even birds have teeth properly for study of the parodontitis. So I'll be talking about one bacterium actually today. It will be Porphyromonas gingivalis. Porphyromonas gingivalis is the bacterium.
Yeah, can you speak a bit closer to
Oh, okay. Okay, Porphyromonas gingivalis is a very interesting bacterium, and it is considered as a keystone pathogen for parodontitis. It has to be there to cause parodontitis. Even at the low numbers, it basically corrupts the homeostatic bacterial flora, bacterium, into dysbiotic, and that triggers the immune response and the disease. And Porphyromonas gingivalis is armed with many virulence factors, but the most important one, which will be the subject of my talk, are gingipines. Gingipines are cysteine proteases, which actually was named after the gingiva and the papain, the end of the papain, and they are expressed by Porphyromonas gingivalis in large numbers. They are the main constituent of this electron-dense layer on the surface of the bacterium. In addition, they are secreted in the free form, like this red pigment around, and also on the surface of outer membrane vesicle. These are the blebs of outer membrane, which is heavily loaded with the gingipines. If you run the SDS page, the major proteins in outer membrane vesicle, OMFs, are the gingipines. They come in two different flavors. One are the cleave proteins after the arginine, we call them RGPs, and second cleave after the lysine residue, and they are the KGP. And P. gingivalis is absolutely essential for parodontitis, the chronic parodontitis. Here's a beautiful picture of parodontitis where you have, tooth which looks pretty healthy, but if you with the X-ray, you will see there is no alveolar bone. There is no bone around. Such tooth will just fall off. You can pull it with your own fingers, you can pull it out of the jaws. And this is a much worse picture of parodontitis with a lot of plaque on the surface, but basically, as you see, the gum line is gone, the bones are gone. And parodontitis is one of the most predominant, prevalent, infection-driven inflammatory disease, which is caused by the Porphyromonas gingivalis, and Porphyromonas gingivalis is the main pathogen. It is anaerobic small rod, which grows nicely on the blood agar as a black shiny colonies. The only drawback of Porphyromonas gingivalis is it smells very, very bad. But for us, it is a smell of success because we know what we are growing. If the lab stinks, that means we successfully grow 10 liters, 20 liters, or whatever amount, volume of the Porphyromonas gingivalis. Here you have this vesicle, this outer membrane vesicle blebbing out. And this, not only P. gingivalis is responsible for parodontitis, but parodontitis by itself, and mostly through the gingivitis is associated epidemiologically, clinically, serologically with many different systemic diseases. And one of these diseases is Alzheimer's disease. In case of the Alzheimer's disease, there's many studies which shows, actually many studies on the perspective cohort studies. I just mentioned several of these studies which keep coming. Actually, there's more and more studies showing that P. gingivalis periodontitis is clinically, epidemiologically associated with the Porphyromonas gingivalis. Here are the prospective cohort studies. There are the case control studies. There are some retrospective cohort studies and observational studies. And finally, there are some case control study, and there are some quasi-experimental study. In some of these studies was 300,000 people involved in these retrospective cohort studies. And all of them shows that periodontitis is strongly linked to rheumatoid, linked to Alzheimer's disease. And for example, here it is the, this association is highly statistically significant. Of course, this is the association. Does it mean that periodontitis and the pathogens responsible for periodontitis are responsible also for Alzheimer's disease? That's a question. But definitely, it looks like that individuals with periodontitis have an increased risk of developing Alzheimer's disease and having the Alzheimer's disease progressing faster and getting more severe. If this is compared to the periodontally healthy controls. All these studies were listed on the previous slides, argue for that. But the main question in such a case is there a causative link between periodontitis and Alzheimer's disease? If we have this, we'll get the periodontitis, we will get the Alzheimer's disease. That's a question, actually. So, in other words, of course, applying to Porphyromonas gingivalis and keystone pathogen in periodontitis, is the Porphyromonas gingivalis a etiological factor responsible for development of Alzheimer's disease? That's a very interesting question, actually, because if it is a case, we can prevent or treat Alzheimer or prevent progress of Alzheimer, eliminating Porphyromonas gingivalis. We know how to deal with the bacteria, actually. So to show that P. gingivalis indeed is associated, not only, but in a causative association, I applied to analysis the Koch postulates. I don't know, some microbiologists hopefully knows the Koch postulates. Koch at the end of the 18th century set up 4 criteria to establish a causative relationship between the microbe and disease. So these are this classical Koch criteria. He applies them to show that that the many severe like cholera at that time, where bacteria were responsible for the diseases. So microorganisms must be present in abundance in the sick people or sick organisms, but should not be present or present in very low numbers in healthy organisms. That was the first postulate. Here is a sick animal, healthy animal have no pathogens there. The microorganism must be isolated from the disease organisms, culture and get a pure culture from it. Use the pure culture to infect experimental animals, and then again isolate the pathogenic organism and show that it's exactly the same as the one isolated at the very beginning. There are the 4 classical Koch postulates. First postulate: the organism must be present in abundance in all organisms suffering from disease, but should be absent in the healthy organism. Was recently replaced actually with the molecular Koch postulates, because they were not looking for the live organism, but if we find a DNA sequence specific for the putative pathogen, which is present in infected individuals, but not in the healthy ones. And in addition, in the anatomical places we affected by disease, okay, this will fulfill the molecular Koch postulate 1. All right. So in case of the Porphyromonas, we will be looking for DNA sequence which belongs to the Porphyromonas gingivalis. Whatever the sequence is, it is but specific for Porphyromonas gingivalis, which should be present in central nervous system, in the most, maybe not all, but the most Alzheimer's disease patients. So in our studies, we have analyzed the cerebrospinal fluids, CSF, and saliva from clinical patients with Alzheimer's disease. As you see, most of them was a clear DNA derived from the Porphyromonas gingivalis. As a control, we use the Helicobacter pylori. There was absolutely no Helicobacter pylori in these patients. So this was in CFS, that was definitely DNA derived from Porphyromonas gingivalis. And if we compare to the saliva from the same person, it looks like that those which have the P. gingivalis in saliva have the P. gingivalis also in the brain. That means the surrogate marker, the DNA from Porphyromonas gingivalis. So, do you agree with yes or no for the first postulate? I hope that the thumbs up. First Koch postulate, yes. DNA from Porphyromonas gingivalis is present in the brain of the Alzheimer patients. Okay, let's go to the postulate number 2. Must be isolated. Nobody isolated Porphyromonas gingivalis from the brain, but this is a very Koch postulate. It's very difficult to prove, because there is, for example, Mycobacterium leprae. You cannot isolate Mycobacterium leprae in pure culture. We know it is a pathogen, but nobody managed to get it in the pure culture. So this even Koch already realized that the second postulate is very difficult to prove. And therefore, recently we replaced this classical postulate with the molecular postulates, molecular Koch postulates, applying for P. gingivalis. P. gingivalis specific antigens should be absent or at least present at significantly lower level in the brains of mentally healthy subjects than in the brains of those with the Alzheimer. So to look at this, how this postulate works, there was the cohort of the patients, more than 100 patients. There was the spacing of the patients. Okay, that's periodontitis patients and sex and age match healthy controls who died from any other reasons. There was the analysis of the MTG of the AD patients and mentally healthy control. There was the immunohistochemical analysis using this tissue microarray, looking for the presence of gingipines. As you know, these gingipines are the major virulence factors. Yeah, it's a lot on the surface. So we're looking for the immunoactivity of gingipines. In the same time, the same samples were analyzed for tau load. Tau tangles are the hallmark, pathological hallmark of the Alzheimer's disease, and also ubiquitin is also highly increased in the brain from the Alzheimer patients. And what you have seen actually, so you have the healthy controls on the top, and Alzheimer's disease on the bottom, looking at for RGP and KGP. Look at this, almost nothing in healthy control, but in the people who die from Alzheimer's disease, there is a lot of staining. And gingipine exhibited very classical punctual intraneural staining in the tissue from Alzheimer's brains. And 96% people have the 51 out of 53 of Alzheimer's have it actually. The same similar numbers, KGP was detected. And it was a statistically significant higher amounts of the gingipines in the brain of the Alzheimer patients. There was some in the healthy old people, which is not surprise because, periodontitis is associated with the age. And, but every one of us here has the porphyromonas gingivalis, the low numbers. It can increase in the advanced age and probably somehow can make it into the brain, but it was much, much less in the healthy brains. And they, gingipine presence correlated very well with the tau load. The red is Alzheimer, blue are control. As you see, there's very good correlation, very strong, statistically significant correlation between the RGP load here and KGP load with the high statistical significance. Also, gingipines, the presence of gingipines and they load correlated also with the ubiquitin load. And the correlation was also pretty strong. So there was a pretty nice correlation with these pathological markers of the Alzheimer's disease in the brain. And as a control, we compare the presence of RGP and KGP in the brain because they are coming from the same bacterium, isn't it? So we should detect them both. And indeed, there's a very good correlation between RGP load and KGP load in the brain, highly statistically significant. The Spearman factor is 6.1. So it is a very, very high correlation, convincing us that these results are true, that indeed gingipines, antigen of gingipines is in the brain. But Alzheimer's brain, several brains from the people who died from Parkinson's disease, amyotrophic lateral sclerosis, and Huntington disease were also analyzed. But as you see here, there was no difference between in the amount of the immunostaining between the load of gingipines between the patients in this case. So gingipine was specific for the periodontitis, for the Alzheimer's brain. And then here it is a very nice immunohistochemical staining for RGP in the hippocampus, the different anatomical parts of hippocampus, hilus, pyramidal layer, granular layer, and molecular layer. So here we have this different layer. You can see blue staining. Here is the enhanced picture. So you see a lot of positive staining actually. The staining which was present, of course, in the periodontal gingiva, where we have a lot of gingipines. In people in Alzheimer's disease in the gingiva have the gingipines. And of course, there was a control and the IgG1 control, which was absolutely negative. So we are seeing gingipines in the brain. And if we look much closer, actually, gingipines are associated with the perhelical filaments of tau, these white arrows. Look at here, the merge. And you can see they really associated with the tau. And they are also colocalized. This is outside the cells, and intracellularly, they are colocalized with the beta amyloid plaque. As you see the red, and here there is the colocalization. So gingipines are in the brain of Alzheimer's people. And there is another paper came out in between after that time showing that P. gingivalis LPS specific for P. gingivalis also is present in Alzheimer's brain. So molecular Koch postulate 2 is yes. Do you agree? Yes or not? Will you agree? Hey, guys, please do. All right. So we have the first postulate was there. So the second postulate, yes, molecular Koch postulate. In comparison to more mentally healthy controls, the Alzheimer's brain shows significantly higher number of P. gingivalis derived DNA sequences and antigen levels. So let's look at another two postulates. Postulate 3 and 4. Of course, if you cannot isolate live bacteria from the infected tissue, you cannot study this postulate. So they were replaced with the modern Koch postulate 3. Virulence factors of suspected organism must cause in vitro and in vivo effects that underpin the pathology of the disease. So in the experimental condition, we should reproduce the pathology of the parodontitis, pathology of Alzheimer's disease. Of course, we cannot do it on human beings, so we did it on the mice. So this was the model, oral infection model, is oral gavage model called, that the mice are basically fitted with the Porphyromonas gingivalis, suspended in the sticky stuff orally, and then after the mice was first treated with antibiotics to reduce the native bacterial counts, then 3 times a week for 6 weeks, P. gingivalis were treated with wild-type P. gingivalis, and P. gingivalis knockout strains, which were deficient of KGP and deficient of RGP. There are 2 different enzymes that were deficient of both RGPs. And at the 5 weeks and 10 weeks, the samples were analyzed for P. gingivalis present in the brain using the qPCR. We are just looking for DNA as a surrogate marker for the P. gingivalis invasion to the brain. And this is expressed the P. gingivalis, PG genome copies per 100 nanograms of DNA. And also looking for beta amyloid 142 accumulation in the brain. This is a picograms per milligram of the protein of the sample. And what happens? Look at here. So here we have, after 5 weeks and 10 weeks, this is the mock infection. Of course, there is no P. gingivalis there, so there is nothing in the brain. But you can see that numbers of the P. gingivalis in the brain measured by DNA, it was increased between 5 and 10 weeks significantly, but also P. gingivalis DNA somehow was making into the mice. Okay, if you look at the mock infections here, but look, and this is it was, 8 different mice. Everyone has some P. gingivalis in the brain DNA, but in the case of the mutated strains, deficient of RGP and deficient KGP, there was no DNA in the brain. So what it means? Gingipine dissemination to the brain in orally infected mice is gingipine dependent. P. gingivalis must have this enzyme on the surface to travel from the oral cavity into the brain. All right, so we also look at the amyloid beta plaque formation in the brain. So it's exactly the same model, same mice. Actually, as you see, this is a mock infection, P. gingivalis infection. There was significant increase of beta amyloid in the brain. But in the mice infected with RGP, in the strain deficient of RGP and KGP, there was no difference with the mock control. So that means P. gingivalis disseminates to the brain from oral cavity and causes a significant increase of the beta amyloid in gingipine-dependent manner. Gingipines in vitro can do also quite a good job on the neural cells. Here we have the neuroblastoma cells, which aggregate if exposed to gingipine. So it's a control. This is the RGP, KGP. As you see, there is the aggregation. KGP is far more stronger aggregation, but they synergistically truly destroy the neuroblastoma cells, causing the aggregation. And further to prove that gingipines can do harm directly in the brain, there was the injection of gingipines into the brain, and the brain was stained for the dying neurons using the fluoro-jet staining green. As you see, mock infection, the saline, nothing. Here we have a lot of dots. If the mice get the RGP and KGP into the brain, which looks like that in the quantitative way. So you see very little here and a lot of staining in case of treatment of RGP, B and KGP. Recently, there are 2 GPT chat-generated abstracts. So they are pretty busy. I made them this morning, actually, from our very recent studies. Apart from mice, we use also the zebrafish model. There's a very nice zebrafish model of neurodegeneration. And this is I hope you can see and read, the screens are big so you can see. This is one paper that P. gingivalis, this gingipine disrupt the blood-brain barrier by degrading tight junction proteins. So we are studying this effect. The P. gingivalis gingipines destroys this junction, allowing the proteins, allowing something to get in. So this was the zebrafish, the larval zebrafish was injected with the Porphyromonas gingivalis, Y type and gingipine deficient strain, and we were looking at the junction, which became leaky. As you see, even large molecules goes also through the tight junction. And there is degradation of the occludins and the proteins, which keeps the tight junction functional. And as you also, there is some degeneration of the vasculature in the brain. And that happens without effect on the gene expression responsible for the occludins and the other proteins in the junction. And different gingipines have different effects. The most, as you see, RGPA, have positive but little effect. RGPB was a little bit better, but KGP, the license-specific gingipine, was pretty strong. So the conclusion is that P. gingivalis, using especially KGP, can open the BBB, blood-brain barrier, and let the bacteria or other molecules to get into the brain. The second similar experiment was performed to show that the P. gingivalis and gingipines can induce the neuroinflammation in the brain if infected zebrafish larvae. Similar setup, but we're looking at the different outcomes from the systemic infection or the local infection. Systemic infection was much pronounced effect on the brain, on the function of the brain, activity, especially on the microglia, and also there was no effect on the behavior of the zebrafish. But anyway, the local one was less pronounced, and everything was dependent on the presence of the gingipines. So this is our status actually, but this right now the growing numbers of the publications showing the same on the mice, but also right now there was a paper on using the zebrafish of the model, in which I just listed some of them which were of some of interest. That PG induces the permeability of the BBB barrier and transcytosis just by gingipines degrading this protein, which is essential for the function of the BBB barrier. In another that's quite recently, gingipines degrade the tight junction proteins, zona-occludens, and occludin, including causing the permeability actually. So this is the way P. gingivalis and the other bacteria can escape into the brain. Together with aging, actually, the BBB is not that tight, actually. It's losing its protective barrier, actually. So parodontitis can help a lot, actually, of Porphyromonas gingivalis. Representing the first, there was a very interesting paper. In this paper, this group used the vesicle, extracellular vesicle, collected from mice and human with parodontitis from saliva, and were infecting the mice with these vesicles. And they cause actually symptoms of Alzheimer's disease in the mice, because these vesicles were causing the BBB getting into the brain and were carrying gingipines actually, those which were isolated from the periodontitis patients. So this was a very, very interesting story. And another very interesting story, it was that, that P. gingivalis using these extracellular vesicles. There's a large number of these outer membrane vesicles are shedding into circulation, and actually they can can, go through the BBB into the brain, actually. So this is, but they must have gingipines on the surface. Without gingipines, they don't go anywhere. So this is also was very interesting studies. More of them, actually, of the studies, the gut microbiota, that P. Parodontitis is causing the dysfunction or dysbiosis of the gut microbiota, and there's gut-brain axis to the causing the Alzheimer's disease, which actually is very, very heavily studied nowadays. And the last one here is that the outer membrane vesicle again trigger Alzheimer-like pathologies in through the inflammasome. And many of these mice actually were cognitively damaged. Actually, they couldn't, they didn't move correctly on this test. So there was definitely, these infections were affecting the cognitive function of the mice, which was becoming the Alzheimer mice. But, look at the publication of the journals. Those are not the mainstream journals. Because still there is a lot of reservation that the bacteria indeed are responsible for Alzheimer's disease. Hopefully, one day, you will try to publish something very interesting, and you will get the editor writing back. No, there's nothing new. Come on, so why do you want to publish in our journal? There's nothing new. Everybody knows about bacteria being responsible for neuroinflammation. But right now, I guess the editors don't like these papers, actually. So I guess if you look at the Koch postulate, did I convince you that the virulence factor of P. gingivalis cause in vitro and in vivo effects, which are really underpinning pathology of Alzheimer's disease? There's many in vitro studies actually showing that through gingivalis, Porphyromonas gingivalis, can mimic all the pathology related to the Alzheimer's disease. All right, but of course, there is a lot of doubts. But then there is another, it was the Koch postulate, it was the fifth Koch postulate, added to this classical one. The fifth Koch postulate? All right, look at that. Koch postulate, that information about the microbe should enable design of effective therapeutic and preventive measure to eliminate disease, to treat this disease. And I put this name, Marshall and Warren, Nobel Prize winner in 2005. They got the Nobel Prize for implicating Helicobacter pylori as the causative function of the ulcer disease, stomach ulcer disease. And nobody believed them. If you read the papers, actually, nobody believed them, and didn't help that the Marshall drank the culture of Helicobacter pylori, got the irritation of the stomach and recovered, fortunately, because otherwise he won't get the Nobel. People, there's a lot of reservation until, based on their discovery, the specific antibiotic regime treatment was designed, and it turned out to be extremely effective in treatment the ulcer disease. Before that, the ulcer disease was, different kind of the mixtures of different things, were tried on the people, but didn't help. But all of a sudden, boom, you put the right combination of antibiotics and something, the disease was treated. So the question is, so elimination of P. gingivalis or blocking its virulence factor should prevent disease development or eliminate pathologies underpinning the disease. Okay, so we developed the inhibitor. There was a phenoxymethyl ketone derivatives, this core 1 and 2, which, if you look here, they are very specific for gingipine. They both are very specific. The IC50 was about 50 picomoles, but did not inhibit the sustained catepsin or other host proteins. They are very, very specific and very powerful for gingipines. In addition, these core inhibitors have the very one important feature. they were going into the brain, actually, by crossing the BBB, getting into the brain very well. That was shown both in rats and dogs, as you see. They were just, showing in the brain. So perfect, because we want them to go there, actually, to prevent gingivitis from doing the harm. And so there was the experimental design. So again, we used the oral infection, the oral gavage model. There was the mice were treated with the P. gingivalis, again for 6 weeks. But in the 5 weeks, there was treatment with the inhibitors, the core inhibitors. And after the five weeks and after 10 days of treatment, we look what happens to the mice. Look at the dissemination of DNA, or let's say P. gingivalis, into the brain. So MOG nothing. After 5 and 10 weeks, look, the inhibitors knock down the level very, very low, to very low level, actually. This is under 10 weeks, all this here. So different core inhibitors were even better than moxifloxacin, the strong antibiotic, which actually kills Porphyromonas gingivalis. So these inhibitors prevented the P. gingivalis, invasion of P. gingivalis into the brain. But not only that, actually. If you look at the pathology, actually, so here we have the, some kind of the summary. So the P. gingivalis invasion of the brain was limited in the concentration-dependent manner, but also formation of beta amyloid was reduced, also in the concentration-dependent manner. Neuroinflammation was strongly reduced, also in the concentration-dependent manner. So inhibitors prevented the P. gingivalis getting into the brain and prevented the formation of the pathological changes in the brain. And this was also the inhibitor was proved that, that after injection actually into the brain, as you remember, the KGP-RGP was causing a lot of damage. But if the KGP inhibitor were given to the mice systematically by mouth in the water, this inhibitor is orally available. That's also very important. So as you see, there is nothing here, which was confirmed by the quantitative measurements. So that works. So fifth Koch postulate. What do you think about it? Koch postulate. The information about gingipines indeed enable us to design the inhibitor which can prevent or prevent the Alzheimer's disease. And I think I will give the thumb. It's yes, there was the mouse study, but mouse is not human. That's a problem. The many studies, especially in the Alzheimer's field, aimed at the beta amyloid, were going through the mice like and then were dropping down like flies at the clinical trials on humans. So, but let's summarize what I said today. So first, P. gingivalis DNA and gingipines antigen are present in the brain of Alzheimer's patients. That's dogma right now. Oral infection in mice provoke brain colonization, increased production of beta amyloid, and neuroinflammation in the gingipine-dependent manner. That was confirmed by many, many other research groups. Gingipine inhibition reduced the bacterial load, block the amyloid formation, reduce neuroinflammation, and rescued neurons in the hippocampus. That's our data. Actually, nobody yet used the inhibitors to show it in this way. So gingipines might be the biomarker of neurodegeneration in Alzheimer's disease, or the inhibitors could be tools. They could be for treatment of Alzheimer's disease. And indeed, they went through the clinical trial. I don't know if you heard about this clinical trial, but here it is the paper, the original paper, which we have published in 1999. And together with the Stefan Domini and Casey Lynch, who set up the company called Cortexine. No, the company Cortexime, actually, it was Steve Dominic visiting me in Louisville, and he had this vision of gingivalis in the brain, Alzheimer. He gave a talk, nobody believed him, of course, except me, so we started collaborating. And they set up the company, actually, and this company developed these inhibitors, which actually went through the clinical trial, the GAIN trial. Gingipine inhibitor for treatment of Alzheimer's disease. It was phase 2/3, randomized trial, double-blinded, placebo control. And it was, assessing the safety, tolerability, two different doses of this core 380A inhibitor, orally available in the subject with the mild to moderate Alzheimer's disease patients. The patients with a PET scan, so it was clearly, you can see that there is not yet very advanced Alzheimer's disease. So these people are still functional, still able to do something. So there was no advanced Alzheimer's disease. What the outcome of this study? It was disaster because there was no difference. As you see, two different doses. If you look at this cognitive function, two different parametric analysis, there was no difference. Why? And all the time I was trying to convince Steve and Casey, select the patients which have parodontitis, which have porphyromonas gingivalis in the mouth. If they don't have porphyromonas gingivalis, this inhibitor won't work, or have totally off-target something. They didn't listen. They wanted big cohort. So they got a big cohort. So this was disappointing. And probably, but if you look at the patient, the subcohort, the patient in which the porphyromonas gingivalis was detected in the saliva, and these patients have strong immune response to porphyromonas gingivalis, the inhibitor works actually. The loss of the cognitive function was statistically significantly slowed down. So there was a very, very good result actually, and probably this inhibitor will make into the clinic somehow. But there was another, the nail to the coffin of this project was, it was toxic. After a longer time, as you see the liver damage, if you compare between this. So that's why the FDA terminated this study. That was, for me, I have about 20 or 30,000 of the stocks from the company, which I can use only after this date. They were at one point $220. But then when the derailed clinical trials, they went down, and I had to buy, I was allowed to buy them for $10 or something like that. No, I guess if this went through, I would be the millionaire right now, probably, but I won't see the pauper. All right, good, but to summarize, because it is not the end of the story. To summarize, so extensive convergent data supports the role of gingivalis in the upstream as an upstream driver of Alzheimer's disease. That is clear from this GAIN trial. Second, this is implication of it is something to have the implication of personalized medicine to treat the people who have parodontitis, who have porphyromonas gingivalis. Not everybody, but just personalized medication of these people to prevent the progress at least to slow down the progress of Alzheimer's disease. So this implicates a further research for gingivalis, gingipines, and Alzheimer's disease. And this is not the end of the story. Unfortunately, I am not a player in the going on studies. That's a pity, but the Cortexime disappeared. Another company came out. Casey Lynch set up the Lighthouse Pharmaceutical. This is a Lighthouse Pharmaceutical, the web page. And if you read here, they just recently received $50 million from NIH to continue. using the different inhibitor of KGP. The same, but basically the core molecule is the same, but they hope that this molecule, this inhibitor won't have the detrimental effect on the liver and other. So they are starting the large clinical trials. As I said, I have no input into this company. They will use my gingipines to develop the inhibitors, but I didn't insist to be on patent, which was my stupid thing, because I gave them the structure, the crystal structure, high resolution, 1.5 angstrom resolution of the RGP and KGP with the inhibitor attached to. And based on that, they developed the inhibitors, but was doing the research, but anyway, I didn't think about that I can be the millionaire in this way. Anyway, so they have the patent of these molecules and the trying. So just the last thing, how this discovery actually changed the citations. So if you look here, the Alzheimer's disease and infection, we published a paper in 1919. It is exponential increase numbers of papers about Alzheimer's disease and infection, but also the papers, so that people start looking at the bacteria and viruses quite seriously, that as the factors which can cause the neuroinflammation in the brain, and in neuroinflammation then drives the plaque formation, tau aggregation, and so on. So, but the same with if you look for the papers, periodontitis, Alzheimer's disease, or periodontitis, or Alzheimer's disease and Porphyromonas gingivalis, I don't have the update actually. So they are growing numbers of these papers. I am very surprised, every week I'm reading another paper about the Alzheimer and Porphyromonas gingivalis. So just the last conclusion. So let's know, if Alois Alzheimer had molecular tools which are available right now and apply them to investigate the etiology of dementia, which was using the Koch postulates, this mental malady named after him, Alzheimer's disease, would be classified as the infectious disease, probably periodontitis of the brain, for example. So here it is, many people were involved. There was the cortexine. She is still very active on the field. Steve is neurobiologist right now in San Francisco. Many people from the company. And there was my group in Krakow, actually, who did all the mice studies for this paper, which was up there. So I'd like to thank you for your attention. That's a reminder what to do. And one more thing is very important, that you believe me, you heard that from the horse's mouth. Thank you. Perfect. 15 minutes.
When I was working at Novartis, we're doing a lot of drug development, and this toxicologist leaned across to me one day and just said, "Chris, tox is everything." And so every aspect of drug development, I always think it's the most important, right? But tox will kill, no matter how good the preclinical data is and the mouse data. You got to do it in rabbits and dogs. And but you need gram quantities of the compounds, and so drugs get killed then. That was a really nice story of drug development as well as the pathogenesis of this disease. One quick question though. So I always assumed that bacteria from the oral cavity from the gingival sulcus enter the bloodstream in a bacteremia when you occlude your teeth. Yes. Do that, right? Is that how you feel about it now, or is it also oral ingestion of the bacteria? Swallowing of the bacteria?
Three different ways, actually. One is through the blood, because that's indeed, if people with the periodontitis, if they brush the teeth, chew on something, they have the transient bacteremia. It's just, it just shows up and disappears, actually. The transient bacteremia, but probably there is enough that some of these bacteria or the vesicles manage through the blood-brain barrier. This is one way. Then the second way actually is trigeminal nerve. Yes, that's correct spelling. Trigeminal nerve. Some of the studies which I listed show that the P. gingivalis somehow can go using this nerve into the brain. So there are two major ways. And then the third one, through effect on the gut microbiota, which are becoming dysbiotic and then somehow affect the Alzheimer's disease. This gut-brain axis, very, very heavily studied right now, and these papers are publishing very well. But parodontitis can contribute to this by affecting the composition of the gut microflora. So these are three different ways, actually, how parodontitis can affect the brain.
Thank you for the interesting talk. I have two kind of naive questions. What do you think if for the prevention, say, if we screen this bacteria in the dental clinic, can we do a prevention for periodontitis and also in the Alzheimer's disease in the future? And the second question is for the treatment to reduce this bacteria in our oral cavity, do you think that can reduce the progression of AD? Or it's like can reverse the disease progression? Thank you.
Yes, there are some papers showing that the periodontal treatment slows down the progress of the Alzheimer's disease. There is a catch here. If people developing Alzheimer, they forget about brushing and so on. So they are just, neglecting the oral hygiene. That's why probably they have more advanced parodontitis. But there was one very interesting paper that indeed oral hygiene, but professional oral hygiene, not only brushing, but professional tooth cleaning, can slow down the progress of parodontitis. So this is one thing, and of course these inhibitors, which like this cortex iron 388 inhibitor, they should mitigate the bone loss or the progress of parodontitis. But in this study, they didn't find it actually. There was no effect on the parodontitis. I don't know. I have no clue why. Maybe it's the pharmacokinetic of this inhibitor that does not finish in gingival clavicular fluid. I don't think they were checking the presence in the gingival clavicular fluid. On the other hand, they used this inhibitor to treat parodontitis in the dogs, and worked. So this is and dog has the Porphyromonas gulae, which basically have all the virulence factors the same as Porphyromonas gingivalis. So it works on the dog parodontitis.
Hi. Correct me if I'm wrong, but I thought periodontitis can have an age of onset, like at a lot of different points in lifespan, but Alzheimer's disease we only see kind of at the end of life, excluding early onset cases. So do you think it just takes years to build up like the amount of bacteria, or is it like a second hit happening later in life?
Both diseases, the main risk factor is age. For everything, age is bad for everything actually, by the way, except for wine. Chronic parodontitis starts developing at the age of 30, 40 or later. So this gap, when you have 20 years before any first symptoms of the Alzheimer's disease, is probably the time where this Porphyromonas gingivalis and other bacteria, the oral bacteria, can get into the brain and start slowly triggering the neuroinflammation, which then turns into the fully blown dementia.
Thank you for your talk. So with periodontitis and Alzheimer's disease, both of those are very complex conditions that have genetic components. Periodontitis has an early onset versus late onset that you talked about, and Alzheimer's as well, early onset and genes associated with that condition. So in periodontitis, there's 500 different species identified associated with the condition. P. gingivitis is one of them that is in, the red group that, maybe, more important depending on what you're studying. But have you looked at a correlation with the genes identified in late onset Alzheimer's disease, about 80 of them, and if any of those genes or pathways are associated and may have a predisposition to interact with the P. gingiva to make it more complex for you?
There is some, at the very beginning, nobody really studied it yet very hard. So just, everybody of us has about 200 bacteria in the oral cavity. If we pull our bacteria together, we get probably 1,000 different species. Fortunately, only a few are parodontitis associated or causing the parodontitis. The other are commensals, which are good to be there. So the problem is actually we still don't know what are the genetic background that some people develop the chronic late onset parodontitis, some don't. Of course, the oral hygiene is very important, but it's not everything. There was a very interesting study in Sri Lanka that, the people don't brush the teeth at all. And still, there was some subset of the population developing parodontitis that are not. And we still don't know that there's some indication. In case of but the same actually a little bit applies for the Alzheimer. There's all late onset Alzheimer that we still don't know except the lipoprotein E, variant 4, which is important, and some other TREM. There are some proteins, but they are not like in the early onset, with exactly presenilin or the secretases, which mutation in those will cause the Alzheimer's disease. And so in late onset, something happened. There are some genes which makes us more prone to develop the Alzheimer. But how did they interact with parodontitis? There was no study like that yet.
Yeah, does any of the ginger pains, do any of them cleave amyloid protein?
Yes, they do. Actually, I didn't show that. Actually, beta amyloid protein was shown to be bactericidal, important for keeping the brain sterile. P. gingivalis degrades that protein.
Does it act as the beta secretase? Because you showed association between A beta 1 and 2 with
it build up in the mice.
This is degradation. Is it going working to get? Does it generate 142?
Yes, it does. It does. It does generate actually directly from the
It's different at each end.
Yes, but it's. There is no arginine, no lysine precisely in this size actually, but what we have shown that it kills the bactericidal activity of presenilin. Presenilin, yes.
So it's indirect.
Yes. But also what it was actually, and also gingipines are causing also tangle formation, tau tangle formation. That was
by inducing the normal beta and then gamma secretase activity.
Yes.
I've got one online question. How do you explain the AD cases without P. gingivalis and all your controls who had P. gingivalis in their brains but no AD pathology? Have you considered that the ginger pains could be just one of many factors disrupting the blood-brain barrier, which could be the actual initiating event for the late causal AD pathology? Okay, so blood-brain barrier breaks down and then the bacteria.
Yes.
I know there's mounting convergence evidence from other known AD risk factors. I think like Mary's getting at age, APO, or genotype indicating blood-brain barrier disruption.
That's definitely not only P. gingivalis. I wouldn't claim actually in my life, but the P. gingivalis is the only one bacterium. Right now, it's more data about the viruses, especially about the herpes simplex virus, which is found in the brain, and then it correlates very well with the Alzheimer's development. Many other bacteria can get into the brain as Porphyromonas gingivalis. And there are some papers, numbers of papers coming out. So there is certainly not P. gingivalis alone which can do, because as we discussed, there's 200 different bacteria, and at least among these bacteria is several different periodontal pathogens which can escape from the periodontal pocket, get into circulation or into trigeminal nerve and get into the brain. So far, we have most data about Porphyromonas gingivalis. It's biased. Of course, every research, this is biased towards. But many other bacteria can probably do the same. But we don't have yet the tools, and then to study everything. And the main problem actually is that you cannot take a sample of the brain, only under the death. Then contamination from the bacteria, for the bacteria, is very easy to contaminate, especially if the body is already for 3-4 days. So this contamination is very, very serious problem. Like, like now, bacteria in the cancer. That, all of a sudden there was discovery of hundreds of the bacteria in the cancer, but this was revoked, they declined these papers because it turned out it was the contaminations. Because the PCR methods are so sensitive that that one molecule of DNA is enough to be amplified. But in case of the Porphyromonas gingivalis, showing the presence of the gingipines, the antigen, also the ALPS in the brain of the human brain is very convincing.
Yeah, I mean, clearly,
but it's not the one bacterium.
Clearly, inflammation is a really common factor, and it's likely different dementias. We call Alzheimer's now are actually a spectrum, and viruses like syphilis and zoster, bacteria like P. gingivalis and other bacteria.
And other bacteria, Treponema denticola, were also found.
So I think on that, unless there's other pressing questions there. No, okay. I'd like to thank you, Jan, for giving us a great talk. Thank you.
All right, so they were good. Nice talk.